Thank you everyone for coming this morning. Before we start, let's take a few moments to acknowledge the sovereign custodians of country, the Aboriginal and Torres Strait Island peoples. Let's acknowledge their continuing presence and their contributions made to both to the Ransco and to the communities in which we work. Let's remember to continue to respect past elders and communities and continue to live, work and interact in a respectful manner with present and future elders and communities. So good morning everybody and welcome to the Glaucoma Update Lecture. And we're very privileged today to have as our speaker Dr. Janie Wiggs, MD, PhD. Janie is the Paul Austin Chandler Professor of Ophthalmology at Harvard Medical School, the Vice Chair for Clinical Research in Ophthalmology, the Associate Director of the Ocular Genomics Institute, and the Director of the Genetics Diagnostic Laboratories at the Mass Eye and Ear. Janie is a true clinician scientist. She started her life as an engineering student at Berkeley, and seeing the way biochemistry impacted people and the changes, switched her major from engineering to biochemistry and did her BA in biochemistry and then a PhD. And in a similar vein, seeing the impact of the biomedical research, then took an interest in medicine, where she trained at Harvard. She did her ophthalmology residency at the Mass Eye and Ear, and then fellowship training both in glaucoma and in medical genetics. And she is board certified both in ophthalmology and in medical genetics. Her research program focuses on the discovery and characterization of genetic factors that contribute to glaucoma, and she is well and widely funded from bodies including the NIH, and very much responsible for a very well-known consortium called the Neighbourhood Consortium, because genetics is a research area that needs a lot of cooperation. So, Janie's qualifications are impeccable. Her research is changing the way we understand glaucoma and will lead to massive changes in the clinical practice of glaucoma for all of us. Her update lecture is called Using Genetics for Glaucoma Risk Assessment and Stratification. Janie.
Well, thank you so much, Paul, and for that great introduction. And just would like to thank the organizers for inviting me to give this lecture, and just really for the hospitality that I've experienced here in Australia. It's been wonderful, and I've had a really nice time. So these are my disclosures, none of which are relevant for today's talk. So our lab and really our field of glaucoma genetics is using a genetic approach to understand the mechanisms of disease for glaucoma. And this basically relies on the premise that the discovery and characterization of genes that cause or contribute to glaucoma susceptibility will identify the molecular events that can cause disease and really also define these mechanisms that underlie disease development. So this kind of information is critical for developing novel therapeutic targets, and that was the talk that I just gave next door for breakfast. And in this talk, I'm going to talk about something that is equally important related to these glaucoma genes, and that's using these genes for diagnostic genetic testing and pre-symptomatic screening, which is critical for glaucoma patients to maximize their available sight. So when we think about genetics in the glaucoma clinic, there are, there's a number of things that we would like to be able to do and to use genetics to do this. We'd like to be able to determine people who are at high risk for disease, and also we want to be able to identify people at early stages of disease when even conventional therapies are more effective. Of course, we want to be able to develop therapeutic approaches at risk, and that's, important focus of this work, but not one that I'll talk about today. So today we're going to really focus on using genetics to risk stratify and to determine disease risk in the populations. Now, glaucoma, I think you probably all know, spans the lifespan. We have, glaucoma affecting children. These are, juvenile onset glaucoma, congenital glaucoma, anterior segment dysgenesis syndromes. These diseases are inherited as we call them Mendelian traits, so they're inherited as autosomal dominant, autosomal recessive traits. They're caused by rare mutations with large biological effects, and because of that, they occur in families. Traditionally, they were found using linkage analysis, and now, we use more modern techniques to find these genes using exome sequencing, genome sequencing. And currently, we have about 10, I'm going to tell you about 2 new ones today, genes that cause these conditions. So in contrast are the adult onset glaucomas, like primary open-angle glaucoma, exfoliation glaucoma, and angle closure. These are not caused by individual gene defects, but are the result of polygenic effects, so an aggregate of common DNA variants that each individually have a small effect on disease risk and susceptibility, but in aggregate, they reach a threshold for disease development and disease risk. Because of the polygenic inheritance, these diseases tend to occur more often in cases than in families. And we find these genes using genome-wide association studies, and we have more than 100 genomic loci that are associated with these conditions. So I'm going to talk first about the early onset diseases and then move into the adult onset diseases. So these are all the genes that are known to cause early onset forms of glaucoma. And they're inherited, as I said, autosomal dominant, autosomal recessive traits. Genetic testing can be done for all of these genes, and there's a real benefit to doing genetic testing for early onset glaucoma. Using just genetic information, we can target surveillance and therapy to people in families who actually have the mutation. We can eliminate unnecessary surveillance of people in these families who don't have the mutation, which is actually a great benefit to the families. This work can inform genetic counseling and risk assessment. And then some of these genes, things like PAX6, actually have some systemic comorbidities that are helpful to know about. I'm just going to show you one super quick example to kind of illustrate this point of genetic testing. This is a small family, but the mother, who's pointed there by the arrow, was diagnosed relatively early at around age 40, although she said she had had glaucoma for many years. And she had surgery to control her intraocular pressure. But she had 4 children, and she was worried about their risk. So we did genetic testing and found that she carried a mutation in myocilin, one of the early onset glaucoma genes. So we tested then all of her children, and we found that her oldest son had very early potential evidence of disease. His pressure was slightly high, but his optic nerves were normal. But what we could do for him is initiate a surveillance plan and even potentially start treating his pressure, since we know he really has this mutation. Her younger son also had the mutation, had no evidence of disease. We could also initiate a surveillance plan for him. And then the 2 daughters did not have mutation, did not have disease. What we could do for them is reassure them that their risk of disease development is no greater than that of the average population. So when we can give this kind of information to families, it's fantastic. The families are really happy to get information about individual disease risk among their children and relatives. But the problem that we have is that with our current set of genes that cause these early onset conditions, we can only give this information about 20% of the time to families. So we really need to identify new genes and genetic causality that is responsible for the remaining individuals. And because these genes had been previously identified primarily in in US or European Caucasian populations, we hypothesized that if we looked in other populations, we might be able to find additional genes for these early onset conditions. And so the first study we did, we carried out with affected families from the Philippines, which is a very interesting, ethnically diverse community. So this is work done by Ryan Calantes in my lab, and we recruited 12 families for this. A lot of people, these are big families. And we did what's called whole exome sequencing, where we sequence all the coding region of the genome. We looked for mutations in genes that we already know cause these conditions. So, and we found some that already did. We found 3 families with mycelium mutations and 1 with a PAX6. And then we applied the sequence data to a pipeline for a bioinformatic pipeline to identify DNA variants that are likely then to contribute to disease. And interestingly, 3 of these families had mutations in a gene called EFE-MP1. Now these are the families, so 2 of them were gigantic families with clear autosomal dominant inheritance, and the segregation of disease with mutations in this gene was a great statistical support, provided great statistical support that this gene was really the causative gene in these families. We looked at the mutations and found that they were, what we call highly evolutionarily conserved. So these mutations affect important parts of the protein in terms of function. So we actually know quite a bit about EFE-MP1 and eye disease. This is also called fibulin 3. And interestingly, this is the causative gene for a retinal disease that's called Doyne's honeycomb dystrophy. There's a single amino acid change, a single missense change, in all patients that had ever been described with Doyne's honeycomb dystrophy, the arginine 345 tryptophan. Also, previously, 1 adult onset primary open-angle glaucoma family had been described with a rare, similar to what we found, EFE-MP1 variant. And then in some genome-wide association studies, common variants in the genomic region that includes EFE-MP1 have been associated with intraocular pressure and adult onset glaucoma. But this was the first report of EFE-MP1 mutations in early onset glaucoma. So to gain a little additional insight, first of all, we showed that indeed this gene is expressed in the anterior segment of the eye in a similar way to other genes that cause early onset glaucoma. So in fact, it's not just a retina gene. It does have impact in the front of the eye. And we also looked at the mutations that cause early onset glaucoma, and we found that the severe mutations that cause juvenile glaucoma cause increased intracellular protein retention. And the retention of these 3 mutations we found in the juvenile glaucoma is greater than the mutation found in the adult onset family previously, and also is much greater than the missense mutation that has been found in Doyne's honeycomb dystrophy. We looked at the cells and found that the mutation was retained in the part of the cell where the extracellular matrix is, suggesting that it causes some damage to the extracellular matrix and therefore results in cell death. So this work has led to kind of an EFE-MP1 genotype-phenotype correlation, where we think there's a correlation between how much protein is retained intracellularly and how severe the glaucoma is. So the more that's inside the cell, the more severe the disease, where the protein mutation that causes kind of this intermediate phenotype in the adult onset glaucoma is secreted more than the severe ones. And the Doyne's mutation is kind of a special case. It's secreted almost exactly the same as the early onset ones, but the protein's not normal, so then it forms these deposits in the retina. So this is a new gene for juvenile glaucoma. We think that this causes intracellular protein retention and then causes increased intraocular pressure because of that. I think importantly, we have not seen these mutations in European Caucasian families. So the absence of these, in those families suggests that doing this kind of work in other ethnicities and in other populations is important for detecting, these disease-causing mutations. So I'm going to tell you briefly about another gene where we also used families with different ethnic backgrounds. And this is thrombospondin, which is also a very well-known protein in the extracellular matrix in eyes. So this story started with this family all the way on your left, which is a European Caucasian family that we found in the United States. And it had a very interesting mutation in thrombospondin, also affecting a very highly evolutionarily conserved amino acid, which points to it being an important part of the protein. But we only had this one family in our collection, and so we reached out to our friends and colleagues, Jamie Craig and colleagues here in Australia. And Jamie had 2 additional families that also had mutations in this gene that affected the same amino acid, which is just really an incredible result. So this was so interesting. So we collaborated with a group at Children's Hospital. Haji Fu here is the fellow who did this work. And we made a mutant mouse that had one of these mutations at this amino acid. And the mouse had elevated intraocular pressure, reduced outflow facility, and reduced retinal ganglion cells, all consistent with a diagnosis of glaucoma, which was really confirming these findings that, in fact, this really is a glaucoma gene. Interestingly, the mutant thrombospondin forms protein aggregates in the trabecular meshwork. And so the thrombospondin here is stained with green. And I think you can see that over time in the mouse eye, that these mutant thrombospondin accumulates in this area. So the mutation causes the protein to change conformation and to form aggregates in the extracellular matrix. This was also confirmed looking at electron microscopy, showing that the thrombospondin formed these dense kind of deposits in the trabecular meshwork. And we showed, doing a kind of an interesting in vitro assay, that if we varied this amino acid according to a range of changes, that we could develop a correlation between how much the amino acid destabilized the protein and how much aggregate formed. So this is another now new gene for early onset glaucoma that also really depended on the inclusion of ethnically diverse populations for its discovery. In this case, again, we find that mutations cause the protein to be unstable, and in this case, it forms aggregates in the trabecular meshwork extracellular matrix, causing disease. So this work now brings 2 new genes to our repertoire of early onset glaucoma genes, which will, add to our ability to offer comprehensive genetic testing for these families. But of course, we have many more to find. And it's interesting that both of these genes are impacting the extracellular matrix. There are so many other proteins that we know that are in the extracellular matrix in the trabecular meshwork that present really interesting candidates also for similar phenotypes. Okay, I'm going to shift now to the adult onset diseases. And here, of course, it's important to remember that these genetic variants are those that impact susceptibility to disease. And one of the really big problems that we have with populations at risk for glaucoma is that there are many people in the population who have elevated intraocular pressure, but only a fraction of those will go on to actually develop damage to the optic nerve and be diagnosed then with glaucoma. And compounding this issue even more is that we have some people who develop glaucoma, so-called normal tension glaucoma, who develop damage to the optic nerve without ever having elevated intraocular pressure. So what kind of screening tests can we possibly develop that is cost effective for detecting all of these cases of glaucoma and detecting them early enough in disease that we can actually intervene in a way that will be useful. Well, we believe that we can use genetics to do this, and using genetics will be more cost effective than doing intraocular pressure measurements on all of these people or doing that and combined with, say, OCT measurements or another measure of the optic nerve function. So when we talk about adult onset diseases and genetic testing, it's important to remember that unlike these early onset diseases that I just talked about, where a single mutation is enough to cause disease, in the adult onset cases, each individual genetic variant that contributes to disease has a small incremental effect. But in aggregate, the disease threshold can be reached. So for the adult onset diseases, it doesn't make sense to test a single gene or a single DNA change, but instead to test a population or an array of genetic variants that in aggregate then can influence disease risk. So this concept of the genetic risk score has recently, in the last few years, been expanded to something that we call a polygenic risk score. And the polygenic risk score is a measurement of genome-wide genetic risk. So, risk alleles for this calculation are derived from very large genome-wide association studies that are sufficiently powered that we can actually have a very good collection of genetic variants to test. Individuals are scored really kind of simply based on the total number of risk alleles that they carry. So in this room, we would count up the number of variants that everyone has, and then array according to the population distribution. Some of you would be at 10%, some of you would be at 90%. Well, of course, we're all at 90% for many things, but in this particular instance, some would be at 10%, some would be at 90%, and then of course, most of the population is in the middle. So to develop this kind of opportunity for glaucoma, several years ago, together with David Mackey and really glaucoma genetic leaders around the world, we formed the International Glaucoma Genetics Consortium that included these are acronyms for all the data sets that were included in our large genome-wide association study for primary open-angle glaucoma. And this table just lists the total numbers of cases and controls that we have in different populations, different ethnic groups around the world. So Stuart McGregor and Pulya Girankakani, who are here actually in Brisbane, led the genome-wide association study, which these are the results, the so-called Manhattan plot. And the important thing here is not to look at every individual gene name, but instead to just understand that there are a lot, and a total of 27 independent genomic loci, and that this is the kind of genetic repertoire that we need to be able to develop these polygenic risk scores. So using this kind of genome-wide association study result, then we can form the polygenic risk score and apply it to different studies. Now, how do we know that this is going to be effective for glaucoma? Well, Jamie Craig and Stuart and other colleagues did really a landmark study showing that they calculated polygenic risk score across the population and showed that people who were in the highest decile, so the 90th percentile of the polygenic risk score, people with the greatest genetic burden for this polygenic risk, had a 15 times higher risk of developing glaucoma compared to people at the bottom of the distribution. So that's really a phenomenal risk effect. And he went on to show also that people in the highest genetic risk profile also develop disease at a much younger age, 5 to 10 years younger than people in the lower genetic burden groups. They had thinner nerve fiber layer thickness, so more severe disease, and they also had a much greater need for incisional glaucoma surgery for treatment. So definitely the polygenic risk score for primary open-angle glaucoma is a tremendous asset for risk stratification. We can find people who are at high genetic risk for disease, earlier onset disease, and treat them earlier and potentially more effectively. Our group has also shown that people with high polygenic risk or high genetic burden have other modifying effects with things like environmental risk factors. So we showed that people who drink a lot of caffeine, which I am one, but people who drink a lot of caffeine don't have glaucoma unless they also have a high polygenic risk score. So the combined effect of drinking a lot of caffeine and a high polygenic risk score increases the risk of developing disease. And we also showed in an interesting study that patients who have a myocilin mutation, so one of these mutations that cause early onset glaucoma, if they have the myocilin mutation and have a high genetic risk, or defined by the polygenic risk score, they have higher disease prevalence and more severe disease than just having the myocilin gene by itself. So again, the combination of one gene insult and then the background polygenic high risk results in a more severe disease. So just to summarize then, genetic testing using early onset glaucoma genes can certainly identify carriers of high impact mutations, allowing for surveillance of these individuals and family, and also allowing for early treatment. Using geographically diverse families, we have found 2 new early onset glaucoma genes, EFEMP1 and thrombospondin1, both impacting extracellular matrix and trabecular meshwork, and certainly providing a path to finding additional genes that can contribute to these conditions. Polygenic risk scores can clearly define individuals at high disease risk and individuals who are destined to have more severe disease. And pre-symptomatic polygenic risk stratification could certainly also be applied for increased surveillance and earlier treatment. So what else do we have to do? Well, we have a lot to do in this field. We still really need a better understanding and a more comprehensive understanding of the genetics of early onset glaucoma. Further gene discovery, I think, is really important to be able to offer comprehensive genetic testing for these individuals. We are also involved in a very important big consortium project to annotate all the known genes that can cause early onset glaucoma and all the mutations in those genes. In Australia, this is an effort led by Emmanuel Souza, and it's really just a tremendous opportunity to understand known existing genetic contributions. We're also involved in a large whole genome sequencing project together with Jamie Craig to identify novel genes that cause these early onset diseases. And we're also interested in really the impact of polygenic risk and polygenic effects on these early onset glaucoma diseases. So, we also need to do a lot more work to fully understand the clinical outcomes of polygenic risk for primary open-angle glaucoma and also other types of adult glaucoma. We want to know about the clinical features that are associated with high polygenic risk, what factors can modify the polygenic risk score. People who have high polygenic risk and don't develop disease, why is that? Or people who have low polygenic risk and do develop disease, why is that? And then we need to understand also how polygenic risk affects risk of family members. What is the family risk of somebody who has a high polygenic risk score? So I'd just like to acknowledge people that have contributed to all the work that I talked about today. These are members of the Neighborhood Consortium. This is our great international glaucoma genetics team with very good representation of the Australians here. In particular, working with us have been Jamie Craig and Puya and Stuart McGregor, and then our other collaborators and funding. Thank you very much.
Thank you very much, Janie. We've got maybe just a couple of minutes for questions, and then I have a certificate to give you.
Oh, boy.
I've got one that's come electronically, while somebody can find a microphone for Hugh.
I've got a loud voice.
Okay, Hugh.
You can use one.
Janie, it's terrific to have you here, and it was a great lecture. Thank you.
Thank you.
I was really impressed with the work you've done on your two early onset new genes and the structural changes or functional changes that follow that. Is similar work being done on the polygenetic changes for in adult glaucoma? How does, if I have half a dozen high-risk genes, how does that actually give me glaucoma?
That's such a great question, Hugh. Thank you, and thank you, for the hospitality in general of Australia. It's been fantastic. Yeah, these are things that we would really like to know. I mean, the polygenic risk score has been a rapidly developing field. I think even, before we get to molecular information, we have clinical information to learn. So, what does it mean clinically? I mean, Jamie has done some great work, and we know some features about these people, but, are there other clinical features that would be useful to know that would point to, some of the underlying molecular biology? And that is something that we're doing right now, and will be exciting to see in the future, for sure. Yeah, thank you for the question.
What proportion of the genes relate to glaucoma due to high pressure, and what proportion of the genes relate to glaucoma due to sort of neurosusceptibility factors? In other words, people think when they mention glaucoma, the nerves are susceptible to damage.
Yeah, also a great question. So far, our genome-wide association studies have favored people who have high intraocular pressure, because most of the studies that have been combined as part of our collaborative work used high intraocular pressure as part of the case definition. So we have very few, studies in, say, normal tension glaucoma. And I think that for that reason, it seems that many of the genes that contribute to adult onset POAG are genes that influence intraocular pressure. There's quite a bit of overlap there. And we do need more genetic studies on, the other side, the people who have retinal degeneration, optic nerve degeneration at settings of normal pressure. And that's something we're working on. So yeah, more to come on that for sure.
Is there a risk of a lower polygenetic risk score being misinterpreted by the patient or glaucoma suspects and affecting their appropriate management?
Yeah, that's a great question too. I think one of the things that we always worry about with genetic testing is that we're going to give misinformation to people. And so, of course, this is one of the situations where you don't want people to think that they don't have, that they're at low risk and they never have to worry about it. And that's one of the reasons why we're very interested in looking at the proportion of people in the low risk decile and the high risk decile that actually have disease. So, the people who have disease in the low risk decile, why do they have disease? Is that where all the mycelium mutations are? So we're involved in a project where we've defined polygenic risk for large population biobanks, and both at our institution and at collaborators' institution in New York City. And we're calling people in with these, relative genetic burdens for clinical exam. And we want to identify, and then further genetic testing, because we want to know exactly the answer to that question. What are those low polygenic risk score people like, especially the ones that do have glaucoma? Yeah. I think right behind you, yeah.
Are there clusters of genes that are protected? Are there clusters of genes that are protected?
Yes, there are. Yes, and in my prior talk this morning, I talked about a couple of those. So there are some genes that are protective. And in fact, of course, could be, genes that are present in people who have high polygenic risk, and then they don't have disease because they have these protective risk variants. And so, that is another important topic that we need to understand more about, especially as we emerge into this era where people are going to be having polygenic risk testing, and we need to be able to give real information back to patients about their absolute risk.
Perhaps just one more question, because we're running very short of time.
That's an interesting topic. Maybe we can catch up on that at the coffee break. Yeah, UV exposure is one thing, yeah.
Okay. Look, thanks very much, everyone. Obviously, it's a fabulous talk, and we'd love to keep going, but we do have another talk coming. So can I thank Janie so much on behalf of the college.
Well, welcome everybody to the Innovation Symposium. For those of you that are hanging around, thanks very much. Andrew, grab a seat. So why are we having this symposium? Basically because we're interested in how innovation occurs, and obviously it's important for patient care. But I think it's a mindset, it's a cultural attitude that we have on a day-to-day basis about trying to improve things for our patients. And also because it's fun, and there's good evidence that solving puzzles, doing crosswords, being innovative actually releases dopamine from your brain. And so we end up getting a sort of sense of satisfaction from solving these problems. We have a panel today which might never be repeated. We have two of Australia's, I think, most experienced and possibly greatest innovators in Graham Barrett and Minas Coronia, who'll be dialing in. We have Jackie Belts, who is an innovator in surgical education. We have Jill Hopkins, who's our international innovator, and Andrew Batty for translation and commercialisation. And we're going to try to explore a couple of themes about innovation and try to get more people interested in it, not just thinking about their cataract surgery or retinal surgery, but thinking about how to innovate and how to be innovators. And when we think about inventions, we often think about the wheel or the light bulb, but I like to think about that first surgeon who performed a couching procedure and how he came up with, or he or she came up with the courage to stick a needle in someone's eye. And there's a difference between invention and innovation. Invention is coming up with the idea, but innovation is about improving it and making it useful or better. The wheel is a good example, but in cataract surgery, we know that to move from couching through intracap and extracapsular cataract surgery to phacoemulsification, there's been a series of innovations where people that we don't necessarily remember have contributed to. And Thomas Edison is actually only one of 20 people who invented the light bulb at around about the same time, but he's the one that we remember. And why is that? It's because he was innovative and he actually worked through the problem of what the filament should be that would actually make it of practical use. He's often quoted as saying that innovation is 1% inspiration and 99% perspiration, but it was actually the 100 or so workers that he had, engineers and technicians, who did all the perspiration. So he was an interesting character, but I think it's a good example of how invention requires further innovation. And Isaac Newton also is famous for saying that if I can see further, it's because I stand on the shoulders of giants. And he's acknowledging the foundations that previous people have made to the contributions that he gave. And I think that that's an important theme, the concept of the Eureka moment. may be true, but we're always building on what people have done before us. And my favourite analogy is the coral reef. A coral reef doesn't just exist. There are a whole lot of little coral polyps that live and they die, and then their skeleton actually builds and makes up the coral reef that we see today. And when we look at this beautiful picture, no one thinks about those coral polyps that have died hundreds of years ago. So is that eureka concept, is it a myth? I mean, do we get that direct inspiration? So Archimedes allegedly was sitting in a bath when he discovered how to work out the density of gold, and then he jumped up and ran buck naked through his village back to his house. It may have been an excuse, but that concept of the light bulb moment, I think it's an important one to explore. But there is some evidence that innovative entrepreneurs possess a creative intelligence, and there's evidence that they use both sides of the brain, possibly more than other people. There are ingredients for the secret source of innovation. These images here are Australian deliberately, because I think that innovation thrives in freedom. You need to have the freedom to ask the question, to explore the question, and places like Israel have a very, very high percentage of patents that are submitted in all areas of endeavour. And I think that's really important. And Australia and the Australian government now are realising that and are investing significantly in innovation and translational ideas. There has to be the meeting of minds and ideas. And magpies, for example, will be able to solve problems more effectively if they're working together than if they're on their own. And often it's organic timing that's important. The light bulb, as I said, 20 people came up with the concept at around about the same time. And the boomerang, we think as being Australian, but there's good evidence that that came into being in various parts of the world at the same time in Europe and the Middle East. And then, of course, there's serendipity. Stephen Bradbury, one of my heroes, who won Australia's first gold medal by making it to the final and the other 3 people in front of him crashing out. So often there's a lot of luck involved in success. Is there a recipe? We'll talk about that. Defining the problem, asking why, connecting ideas, observing, experimenting, collaborating and communicating. I think that they're important themes that we need to flesh out. So without any further ado, we will get the panel moving. I'd firstly like to acknowledge the panel leader, Jackie, who will be leading the discussion. And as you know, Jackie is a corneal and cataract and refractive surgeon from Melbourne. Her innovative thinking has resulted in significant change to surgical training in Australia. She founded an education program called Geni, and she's an entrepreneur in beauty and digital health industries. And she was recently made co-president of the Australian Society of Cataract and Refractive Surgeons. So thank you for coming, Jackie. And our first speaker will be Graham Barrett. So Graham, as you know, is a consultant ophthalmologist from Western Australia, and he's a clinical professor at the University of Western Australia. His area of interest and expertise are cataract, implant surgery, corneal and keratorefractive surgery, with particular reference to lens implant formula, which I'm sure most of us use. He's received many medals, including the Ridley, the Binkhorst, the Kelman, and the Barricare Awards. And he's still on a number of boards around the world, including the International Society of Refractive Surgeons, and he was the founding president of Oscris, as you know. And in 2021, what a lot of people don't know is that he was given an Order of Australia. So Graham Barrett, thanks for coming.
Thanks, Jared, and thank you for putting forward such an enterprising symposium. What an interesting topic to discuss. And Jared, show you my disclosures, but they're not relevant, or they're all relevant, I'm not sure. But it's a fascinating topic, and thank you for asking me about formulae for innovation. You can interpret that in several ways. And what I've chosen to interpret this as a series of cases that we can then use as examples to have the most interesting part, which will be the discussion. But when I think about innovation, and I think you're going to get different individual ideas on it, for me it's about problem solving. And personally, I think my stimulus for innovation is problem solving. And the problem which I was faced with when I began cataract surgery in 1980, that this beautiful operation wasn't beautiful enough. And my efforts have really been about trying to make cataract surgery safer and more predictable. So how to begin discussing innovation? And if I look back at my own efforts, they very much are about new procedures, changing techniques, improved technology and innovations. And when I have an idea or innovation, I tend to make a film about it and submit it for a film festival. So I'm going to share a few reels from the shelves. And the one theme, if I think about innovation, is, for me, has been reducing astigmatism. And the first reel or theme I'll share is this video, which is innovative in that we hired the Supreme Court room in Perth, and it talks about a surgeon who had to defend himself in court because he'd left one diopter of residual astigmatism. And this was a vehicle to talk about a fascinating innovation, which is this keratoscope. It's a quantitative keratoscope that you look down at the cornea, and at a glance you can tell how much astigmatism there is at about 1/2 to 1 diopter. I think it's one of the brightest ideas I've ever had. And I use this as a case example of what I think is a wonderful idea. And to make it happen, I had to then develop injection molding and produce it myself because nobody else wanted to. And it was available somewhat through Duckworth in Kent, but I used to supply them the molded keratoscope for free. And you've never heard of it. And so the interesting thread, I think, through the discussion is what takes a great idea, because I think this was, and what makes it something that actually gets used and is successful. So this is an example of a wonderful idea and remarkably unsuccessful in terms of taking this into something that anybody else has heard about. And that was not inducing astigmatism. And the next video I'll share with you was about reducing pre-existing astigmatism. And that was looking at, when we do cataract surgery and we use toric lenses, how can we predictably reduce astigmatism? And my last words in this video are, don't blame the lens, blame yourself for not using a reliable method of predicting astigmatism. And it was called Flight of the Arrow. So there's no device, but there's a lot of thought gone into the problem and solving the problem of predicting residual astigmatism. And you can see each area of thought leads to another one. So the keratoscope leads to toric formulae. And then, of course, the next problem I faced with astigmatism was the alignment issue. I couldn't stand doing predictable formulae and then aligning it just by eyeballing the eye and putting some blue dots on it. And this was an insight that within our iPhones, we actually had a wonderful device to determine the level, true horizontal level, and deal with cyclotorsion. And this led me to making an app called ToricCam, which I still use. I do use image guided, but I always use the app as well because I think it's more reliable. And this case example is something we'll talk about, but when you have an idea, to make it happen takes a lot of effort. So I have a smattering of coding. I'm just self-taught from about 1981, but I'd never made an app before. And, you know, after 6 months of hard work and reading books, I managed to make an app and put it on the App Store. It's a huge negotiation to get something on the App Store. And the message here is, you know, don't be daunted. Don't know your limitations, because I would suggest you don't have some. You don't have many. But let's put these case studies on astigmatism to bed. And this is not chronological at all. And talk about, perhaps for me, which has been the most fertile area for me and occupied my thoughts more than anything else, which is phaco and IOL, design, fluidics. And this movie was called The Child of Evolution, and it portrayed the development of what was the first foldable lens in about 1983, and tried to predict what the future would be, suggesting that a single-piece lens with soft haptics would be part of our future. And this lens design, which was hydrophilic acrylic, influenced the original one-piece hydrophobic lens by Alcon. It was a precursor, because I was working with Alcon at the time. So we can talk about, perhaps in the discussion, what I think this case study illustrates, which is something else. But I'll come back to that particular thing. So sometimes you do have eureka moments, and this wasn't an excuse to show my bare chest in the shower. It really did trigger my thoughts on how to deal with, as the incisions were getting smaller, they were getting hotter. And this is a microflow needle. I regard it as one of my most successful because I'm still using it today. So this was 96, and wouldn't like to operate without the microflow needle. Another cautionary tale, think carefully about your names, because I called it the MaxiFlow needle, and not realizing this was a widely used sanitary product in the United States. So that's why you've never heard of the MaxiFlow needle, but you have heard of the microflow needle. So some innovation is not about devices and gadgets. It's about how you apply techniques, how do you apply different ways of dealing with presbyopia. And so The Best of Both Worlds is a movie about my obsession with modest monovision as a solution to presbyopia. So it's not anything else but a lot of thought on what were alternatives to multifocal lens. Also an excuse to share my observatory, because besides our lives as ophthalmologists, we have other lives in building this very strange building, and certainly was probably occupied my creative thoughts more than most. And we can take up that thread. Moving on, and you see how one idea leads to another, because the monovision led me to the next idea. And this is what I spent 25 minutes talking about yesterday, and that is developing extended depth of focus IOLs. And the art of Zen and the art of motorcycle maintenance, or monovision, was about extended depth of focus, which, as I explained, you may not realize was really the first extended depth of focus IOL. So the lesson here is this lens you're hearing about because Rayner is producing it as a product, but I'm telling you this was the first extended depth of focus IOL. So 15 years ago, I was convinced this was a solution to presbyopia, and spent the years knocking on doors. And that's how long it takes, 15, 10 years sometimes. And so taking in ideas to eventually useful embodiments which can be used by other people is not a short pathway. Now, having spoken about my first love, which is IOLs and phaco and fluidics, I do have another life, and that is doing corneal surgery and refractive surgery. And I'll share a few activities in that area. So let's take out the role on keratoplasty and refractive surgery. And so this is what I did when I got back from my fellowship in the US. I had a choice. I could do refractive surgery, and in those days that meant doing radial keratotomy, which I decided I didn't want to do. So instead I spent my time developing a technique using corneas lathed on a cryolathe. So it's a new method of lathing for myopic lenticules. And of course it's building, and Jared, you mentioned that innovation builds on the ideas of others, and it's just really Barraquer's keratin mallucis, but using somebody else's cornea to do the lathing on, not the patient's own. Besides that foray into refractive surgery, this was pretty brave, I thought. This was 95, 96, and here I'm inserting a little corneal inlay, 1.8 millimeter, into the deep stroma. And what am I trying to fix again? Once again, presbyopia. And once again, this was another way to resolve this problem without multifocal lenses, inspired by the insect eye with its multiple foci and the final frontier. So almost successful, because when you want to take an idea and make it real, you do need to partner with the industry. That's a fact of life. So choose your partners carefully. I chose Chiron, which was a good choice because they were very enterprising and innovative. But of course, they got purchased by Bausch & Lomb, and in 95, 96, this was far too daunting to bring to the market. But it was, you know, we got quite far along the line. And it's interesting to see almost, you know, decades later, the resurgence of looking at inlays in the cornea. This one actually worked, so the actual material, the location, were all carefully thought about. But that's another thread that we can bring out in the discussion about timing. And if you're too early sometimes, that means things don't always work out. And then finally, Jared, you'll be appreciative, I'll get to formulae. And this movie was called The Myopic Mystery, and it was looking at the results in high myopes and realizing that I didn't get +2 and +1 refractive errors because I was using my own formulae. And so this movie was explaining the reasons why you get hyperopic errors in myopia. But I'll use this as a digression quickly to talk about formulae, because Jared asked me to as well. And you see here what made me interested in formulae. So it wasn't a desire to make the most accurate formulae. I didn't have much interest in that, to be honest. But when I put that first lens in 1983, I had to work out what the power was. And because it was a different refractive index, different material than PMMA, different refractive curvatures, etc., I had to have a formula where I could put these different indices, these different parameters, and that takes you to a really complex formula, which is paraxial ray tracing, where you can individualize all these parameters. So it was, again, an example of problem solving. What was innovative was the ELP prediction. And I always like to try and understand, well, what is behind the fact that more myopic eyes have deeper anterior chambers. So I came up with a model eye, and the model eye simply is that look at the eye as two spheres. It's got an anterior sphere, which is related to the radius of the cornea. It's got a posterior sphere, which is related to the radius of the globe. And that intersection defines a plane which divides the eye into an anatomical chamber depth and a lens factor, which is the distance between that plane and where the physical position of the lens is. And that's the basis of the universal 2. You can't measure the radius of the globe, of course, it's difficult to measure, so you use, in my case, polynomial regression to derive that parameter. And you know, the toric formula is based, again, on universal 2. But once again, there's a model behind it which is trying to explain not what the posterior cornea is, but why the posterior cornea persistently creates against the rule aligned almost always at 90 degrees. So it's a model which then I add on the maths. And it's exactly the same with the True-K formula, which is for post-refractive eyes. The problem is the disruption in the relationship of the posterior cornea to the anterior cornea, the radii. That relationship is disrupted. And the basis of the True-K is basically BU2, but with algorithms to deal with this issue, which I've explained. And that's the reason it can be used for myopic, hyperopic, RK, and more recently with keratoconus. Because this formula is paraxial ray tracing, every time you predict a lens in a lens power, it's calculating where the principal planes are, the first and second principal planes, for each lens that you calculate. Now, this has consequences, and I can show you the Hager's formula, single line of code. I can show you the SRKT formula. I can show you the holiday formula, and Jared, here's the secret. This is the BU2 formula. So in its simplest form, it's about 750 lines of code, because it's more a method of solution than an actual single line of code. And it goes up to 3,000 lines of code in the more complex things like toric calculators. So we'll move on from refractive surgery, and I'll share with you what I've been doing for the last 2 years. It's somewhat related to ophthalmology because it stems from my desire to continue to interact and communicate and to educate while we're all shut down. It's not in the shelves yet because it's ongoing, but let me briefly introduce you to Ivy. So what Ivy is essentially me realizing how poor communication was. Yes, we had Zoom, but the microphones were poor, the speakers were poor, and most of all, we lost eye contact. And so I tried to rectify all these things and really put everything into one device with microphones, with high quality speakers, great video. Webcams are really quite inadequate, but really good camera. And most of all, what this device does is one cable connection to your PC or to your laptop, or you can have an integrated PC as you see here. But what it does, it restores eye contact. So you just look at the people you're talking to, and they will see you looking at them directly, as you see in this example over here. It's a totally different journey because you've got industrial design, you've got electronics development, and then you have to face the expensive questions of tooling and manufacture, and of course we're in the process of doing that. So I've told you what I think is a stimulus for innovation, which is problem solving. But of course that doesn't tell you the whole story, because what is the essence of innovation? So I'll share with you this little scrap of newspaper which I carried in my wallet for many, many years, because when I read it, it really triggered something in me which I recognized immediately. And this is Isaac Asimov. Who's read his books? Yeah, you must have. Wonderful books. Anyway, so in the 1950s, he was asked by the US government to form a committee to discuss this topic of innovation and how to encourage it and how to provide a basis for future American innovation. So he lasted 2 weeks and then he resigned. And before he resigned, he wrote this little essay, which may be a bit challenging, Jared, because I'll share his conclusions. So the first thing he said, innovation is not really something that you can put a committee to. And you can't actually probably even have a panel discussion and try and discern what it is. But he says, it comes from inward reflection of individuals rather than group discussion. And he made a point that it comes from people of considerable self-assurance. Now, what does he mean by that? When I read that, when I was preparing for this talk, because the piece of paper, unfortunately, is disintegrated, but when I looked at the slide, I thought, well, what made a first-year registrar think that he could change something which had been done for decades, which is lenses are made from PMMA, and that's what they should. And not only is he going to change the material, but he's going to make a lens and develop a new lens based on this material. And it's, I'm not sure if it's self-assurance, but it's self-belief. It's a belief, and it may be false, but it's a belief that you can do anything that you set your mind to. And I think we all can. But a lot of us, you know, I think by culture, I think by parents or whatever, that belief gets challenged, and so it does take, and you know, when I see self-assurance, that does ring a bell. This is only after reflecting after many years, of course. He also made another point that the world in general approves of creativity. He said these people are somewhat eccentric, and he said even to speculate in public is rather worrisome. So I'm going to ignore that caution, and I will speculate on the essence of innovation and give you my thoughts. So I think the key thing, we spoke about motivation, which is problem solving, but the key thing of innovation to me is curiosity. And what is curiosity? Well, we've all got kids and we've got grandkids, and sometimes you need grandkids because you're more reflective when you have your grandkids, and you watch those little kids and you watch their curiosity, and they want to try everything. And you know, what's the question your 3-year-old keeps asking you? Why? Keeps why. Unfortunately, what happens as the kids get older and you watch, and in ourselves, that curiosity tends to be dampened. We tend to lose that curiosity. So my thesis to you is that creative people tend to maintain that curiosity and questioning of why and how can I make it better. But Jared made a good point. We all need inspiration, and our colleagues feed into that inspiration. And there's no doubt that the discussions, the talks, the meetings, the coffee breaks, all those things help innovation. And I think a mentor is excellent, and I was very lucky to have 2 great mentors in that process. But besides those colleagues, the contact with young, interesting minds is also very helpful. And I've been very fortunate to have many, many fellows over the years, and that really helps my own sense of innovation and inspiration. And the last element is persistence. The ability to persevere, and you mentioned Edison, I think he picked up on that one, and I think it's real. It's tough to take ideas to reality is tough. It takes persistence. The only secret source I have for that, in my own respect, is the support that you get from those around you. And for that's my wife, and not only my wife, but my kids. And just to show you how they support and participate, that was this year at Oscars when we handed over that leadership to Jackie and to Jared. And you know, just to have that support keeps you going. Don't ask me the secret for a balanced life, because I don't think there is such a thing, not if you want to innovate. But what helps you keep you balanced and keep you persistent is your family. So thank you for your attention.
Thank you, Graham. We might just get Minas to talk, and then we'll have a discussion about some of those concepts and ideas, and hopefully, or possibly some disagreement about that. But Minas, as you know, is the chairman of the Department of Ophthalmology at the University of New South Wales, based at Prince of Wales Hospital. He has an international reputation for research in cornea and ocular surface disease, cataract and anterior segment surgery. He is an authority on the pathogenesis and management of pterygium, and is an inventor with over 30 issued patents, including Vision Blue and the core patents for the microstent CyPass. His work has also been recognised with an Order of Australia. So, Minas, I know you're out there somewhere. Come in, please, and present your lecture.
So, Jared, can you hear me and see me?
Yes.
Okay. So, thank you for inviting me to be part of this, what is in fact an innovative session. Virtual hello to other panel members and to friends and colleagues. This is going to be a more sort of nuts and bolts presentation as compared to Graham's, but here we go. I added the question mark to the title, and you'll see why as this goes along. So this photo was taken almost at gunpoint, so it's probably worth a second look just to make a few points. It gives you an idea of perhaps how I think. Most of us are going to have the privilege of working in a dark room using a slit lamp for most of our careers. We'll have the satisfaction of making people see better, even saving lives. will earn a comfortable income and have good job security. The downside is that we're likely to become vitamin D deficient, which carries some mortality, get sore backs, and be exposed to viruses that can damage our eyes and even kill us. Mostly we are there to fix things in front of us. There are a lot of things we don't fix all that well or at all. For the things we can't fix, we should be thinking of solutions. And so we're better able to treat entire populations. And the best ideas I've had have come from sitting behind that machine. The third point is that since sight is critical, an early metaphor for opticians was to help society see better. So no big surprise that there've been a number of ophthalmologists involved in politics from the early days. Teaching this approach, I suspect, is best done by setting an example. And Fred Hollows was always on the hunt for better ways of, mostly pragmatic ways of fixing things. I tried to continue that ethos in our department. He also recognised, and Graham has touched upon this, that change isn't easy. People don't want to hear that their approach is wrong, and even sometimes downright stupid. The slit lamp is a great example for much of what is wrong with our approach to innovation. So there's been little change in it in over a century. There's been a focus on the optics with little thought as to the health of the operator. So long as the optics are fine, who cares that 90% of you will complain of sore backs? And that by being 30 centimetres from the patient's face, we're prone to infection. Even the breath shield was only added in about 1920. So it might be a bit difficult to see, but here I have a laminar flow fan in the workspace of my slit lamp. I can actually open my window if I feel like it, and I've got a souped up aircon system. And I'm sitting in a specially designed chair which redistributes weight from my lower back to my knees. So companies that make and sell slit lamps often don't actually listen to what we have to say. And some companies will defend a position, for example, making intraocular lenses that opacify or cause dysphotopsia. They just hire a better marketing team. If you then propose a solution, they may even steal it and challenge you to take them on. A patent is only as good as your ability to defend it. As doctors, we come from a different planet. We're about caring and healing. In the business, and in particular the venture capital world, you eat what you kill. It takes a bit of getting used to, and this attitude infects others around us. Graham is right. By working in teaching institutions, you get to be challenged by very bright, but sometimes difficult people. You get the best out of humans when they spark off each other, and I've had wonderful experiences with this. But you soon work out that you don't, and you soon work out that you don't know everything. You can't know everything, and in a way you get a sort of a prismatic effect of looking around a problem from different vantage points. A big issue is to work out when collaborations benefit all parties. In game theory, the prisoner's dilemma. If you collaborate with people that use the conventional pathways of research, adding a tiny slice of extra knowledge to an existing paradigm, often turn out to be wrong. If it was that easy, a solution would have been found earlier. Don't hunt with the lemming pack. Frugality is a good indicator of innovative ability. Some of the best surgical videos at the ASCRS Film Festival are from India, a well-educated workforce, and sometimes they have few resources. In 1995, my laboratory was closed down, and I've existed without one ever since. It turned out that this had great advantages. I haven't been weighed down by the day-to-day managing of a laboratory, nor dealing with issues of bright and sometimes not so bright people with huge egos. Secondly, dealing with universities and hospital administrators where money is concerned really is an unedifying task and wastes a lot of time and energy. I was once asked by our institutions how to foster innovation and invention, and I said that you take away all of the resources, and if the researcher survives in their field, they must be innovative. Another example of this is when I was involved in one of the early bionic eye groups after Kevin Rudd's 2020 summit. We missed out on funding of around $25 million, very hard to accept at the time. But in many ways, I was lucky. I was very pleased to have missed out on the management and human resources issues and failure. In fact, the bionic eye funding is the subject of a PhD thesis which used this as an example of how not to fund biotech in Australia. So I had to become fairly efficient in appropriate collaborations, and I've managed this with a number of overseas groups. And one of the key things was repurposing both of ideas and devices. Finally, on this slide, in an evolutionary sense, what better way to engineer innovation than a crisis that threatens your very existence? In January 2020, I began a review of coronavirus infection. I knew we were going to be in for a real fight, and rediscovered that a major pathway of infection is via the ocular surface. The last 3 years have been the most productive of my career. You'll see here I'm wearing my now favorite color, blue. And so many of you will know the story of Trypan Blue and its development, and my apologies if you've heard some of this before. Fred Hollows, who established the college's National Glaucoma and Eye Health Program in the mid-70s, passed away in February '93. He was always going to be a hard act to follow, and I realised shortly after that we had around 100 patients cataract blind out in Bourke. And we put together a team and carried out the first cataract list in Bourke. And here is a very young team, a young Jeff Wilczek, Alan Flax, Jason McCabe, who put together all the technical side of it, and Joanna Barton, who has run the show for many years. The problem was that by the time we got there, most of these cataracts had turned white. So here I am with this fantastic team from Sydney, a new phaco machine, and patients with very high expectations. This was not good fun. Mother of a US friend and colleague had two sayings that resonated with her son and with me. The first is that need cuts steel. And here clearly was a case of great need, something really worth fixing. And on the 10-hour drive back from Sydney, I was hell-bent on fixing this problem. And the reasons we all know, huge market, numbers of cataracts everywhere. Success is technique dependent. You need to have a good capsular rhexis, and improved visualization was key to this. So how did this come about? Well, as an undergrad, I began research in Professor John Young's laboratory at the renal lab at Sydney Uni. It turned out that I ended up working on salivary glands as a model of electrolyte transport. I learned microsurgery, some cellular physiology, and how to read the literature. Here is a rat with 6 salivary glands, major salivary glands cannulated with little pipettes to measure flow rate. But we had to express secretion in terms of the weight of glandular tissue. And so to identify the glands, we inject a dye, various dyes, either through the duct or into the blood vessels. And so this is my first experience with in vivo dyes. And here is a pet peeve. Those of you that have worked to dumb down the basic science and research requirements for entry into ophthalmology have likely done more damage to harm innovation than you will know, and will ultimately consign Australian ophthalmology into the mediocrity bin. My next encounter with dyes was in the early 1980s when laser trabeculoplasty started in Sydney, and Colin Warner was our senior registrar at the time, and he noted that he got poor results if the eyes were non-pigmented. So we discussed how to fix this and came up with the idea of using a dye that would make the eye more sensitive to the laser wavelengths. And actually this ended up in the original patent for Trypan Blue. So where did I look for this? Well, at the time, Prince of Wales Hospital had a number of factories around it in South Sydney in particular. And we had this book, Grant's Toxicology of the Eye, floating around on a bench somewhere in the department. So under D for dyes, I ran my finger down and came across Trypan Blue. In this book, there was reference to the work of Norn, who did a lot of work with dyes in Scandinavia in the early 1970s. And he used Trypan Blue to stain the corneal endothelium in patients who are undergoing then intracapsular cataract surgery. And almost as a throwaway line, he noticed that the capsule of the cataractous lens became coloured over the area corresponding to the pupil where the dye had access. And he noted that it was the capsule rather than the epithelium. And then very conveniently, he did an 8-year follow-up study to show there was no real evidence of toxicity. So got some dye, got a then very helpful hospital pharmacy to make it up. They, of course, were cautious and warned us of all the things that could go wrong. And we did a small clinical trial. Evan Soicher was my registrar at the time and did a great job organising much of this. And so he was the invention. Those of you that might be curious about the name, it comes from a Greek word meaning borer, perhaps not unlike the speaker. And so the trypanosome, which caused sleeping sickness, is a real problem in the German colonies in Africa. And Paul Ehrlich in the early 1900s was tasked with fixing this problem, and trypan red came out of the research, which actually worked fairly effective in killing the trypanosome. By asking around, I found an excellent, brilliant Aussie patent lawyer, Peter Stern, who wrote patents from the perspective of defending them. Some heavy-duty people looked for flaws in the trypan patents, he wrote, and failed. They were bomb-proof and remained unchallenged to their expiry in 2019. Peter taught me a little about patent law, and I taught him a little about ophthalmology. It was a wonderful collaboration. Peter's firm had contacts in Washington and worked out fairly quickly that if something was held up and there was a problem, you actually went to the patent office yourself and presented your case. On the crest, you'll notice that the eagle is holding an olive branch in one talon and arrows in the other. A pretty good summary of the state of play. So this patent was issued in 2002. We'd filed it in 1999. And in those days, for a US patent, you had to prove precedence. So this was proof that we did this work first. The next thing was to sell it. Someone in Allegan wrote back to me, nice idea, but we don't have such difficult cases in the US. A good thing was that they introduced me to Gav Weifinkel, who had been an instrument maker in the university department in Rotterdam in Holland. And he'd set up Dutch Ophthalmic Research Corporation, and he is a very good person to deal with. I found out at eventually our first meeting, because all of this was done over the telephone, that he had a band as an apprentice, very keen Deep Purple fan, and this is one of the Deep Purple band members' guitar that was given to him as a gift at a function. He very quickly worked out that there had been work on trypan blue in Holland, and by careful negotiation, we avoided a patent dispute and everyone got on and did productive things. So at the first Deep Purple concert, I got red carpet treatment and we've remained on good terms ever since. I was advised not to publish this work, and Gerrit Mellis gets the credit for the first publication. So most of you have experienced this dye and what it can do. In the early days, we reserved it for white cataracts and bad corneas. And pretty much, it made a huge difference to our ability to safely carry out this kind of surgery. So eyes like this, you could simply just take out the cataract and they get great results. So it cut down on the number of corneal transplants we did and triple procedures and so on. It found wide use in other parts of ophthalmology. It is used in DMEC, working out which way conjunctival grafts are, in retinal surgery, even in orbital surgery, you can stain tenons with it. The next part of the story is interesting. In 1988, this great movie came out with Steve Martin and Michael Caine, Dirty Rotten Scoundrels. And it was, you know, a good sort of preparation for what was to come. There's always competition. So some US podium speakers made claims that the trypan blue weakened the capsule based on a few cases. This went on for years, in spite of the fact that we and others showed that this was not the case. You could stain a capsule, paired eyes, do rhexis and compared non-stained with stained eyes. And if you stretch the rhexis, we showed that there was no difference in the force required to cause the capsule to break. In fact, trypan stiffens the capsule, and I think it makes it easier to tear. And these days, or for many years, I've used it on every case that I do. One way or another, this product sold. By 2013, over 4 million units sold around the world. So fast forward to the present, Garrett went on and had many concerts, sold Dutch Ophthalmic, and bought a company called Tool Meditech, which makes the SurgiCube, which enables people to do surgery in smaller facilities. He became an expert in laminar flow and clean air and so on. So when COVID struck, I looked at the slit lamp and thought, this is not a great arrangement. And Bifingle's firm made a first prototype of this laminar flow unit for slit lamp. And if you blow smoke, so to speak, it will get into your eyes and those of your patient. But if you turn the fan on, it'll get rid of the smoke. And so there are various versions of this. There can be a suction version as well, and so on. And we're trying to convince some slit lamp companies to take this on as a more hygienic way of doing business, because after COVID is over, many of you will have suffered adenovirus infections, and these are essentially airborne. As for COVID, the foment's story has probably been overplayed. These are airborne viruses. They're respiratory viruses which are oculotropic. They head for the eye. Another pet peeve is the overuse or misuse of the words innovation, translation and hacking. It reminds me of when Hillary Clinton was at her height, and a lot of mostly American women adopted her middle name as their middle name. A whole lot of presumptions made that there's an ability to think outside the square or cube or whichever dimension you're in, that in order to translate, you actually need a second language from which to translate. And if you don't, for example, in my case, have had a basic science background, what can you translate? And on the weekend, I was meant to have been at a hackathon at the university to hack the COVID pandemic. One of the things about diseases is you can hack away as much as you like. It's mostly destructive, and then they're immune to being cancelled. I thought I'd finish with this video clip from Richard Attenborough. Check out the giant trevalle's eye. I saw this some years back, and we actually started a project looking at light damage in fish. Perhaps we'll find the first fish pterygium.
So there is a fish here that amazingly has a brain capable of calculating the airspeed, altitude.
Sorry, I'll just do that again.
So there is a fish here that amazingly has a brain capable of calculating the airspeed, altitude and trajectory of a bird.
So I suspect that what comes out of the blue is opportunity. It took millions of years to innovate the trevally. Thank you.
Wow, thank you, Minus, and thank you, Graham, for your presentation before that. And what a privilege to be in a room with two great innovators speaking at the end of this Ransko conference. I was wondering if we could start the discussion going right back to something that, Jared, you mentioned in the introduction, which is the innovative mindset. And Graham and Minus, maybe one at a time, and just briefly, you could explain if you think that you've always had an innovative mindset, or if you've developed that over time, and what and who inspired you to end up with this innovative mindset.
Jared, do you want to go first, or?
I think Jackie asked you, and Minus, not me, but...
Okay. Yes, no, of course it's there from your childhood, and you know yourselves if you are or you're not. the child who's taking down radios, never manages quite to put them back together, gets into trouble. Playing with the chemistry sets. It's a dangerous life because you're making gunpowder and you're, blowing up kitchens and doing all that type of thing. And yes, I think it's something you born with. I think we're all born with it, but as I said, most of us tend to lose it, Jackie. That's the secret. What keeps it going in some people?
Minas, can we hear from you if you could hear my question?
Yeah, no, I did. And thinking about this over a good period of time, I can trace my family back to around 1600 on an island just north of Crete. Now, island populations are very interesting because if you didn't have a good genetic background and survival skills, you basically didn't survive in the years before medicine. And part of that survival, the smarter you were, the better you could survive. There were people that built water mills, for example, which gave them power. And my family were involved in one of those ventures, and I've been back to see it all. My family left for the United States in the early 1900s, and we ended up in Australia in the 1920s because migration to the US had stopped. So I come from a family of survivors. At home, growing up in a country town, I had access to a workshop from a very early age. And in those days, instead of throwing things away, you tried to fix them. My father had been in business during the Depression, didn't throw anything away, and I'm still a bit of a hoarder. In the first year of high school, we had 6 months of metal work and 6 months of woodwork. And so I still make many of my prototypes. And so I came from a sort of background of, as I emphasized in my talk, fixing things. And Jared is probably right. There's probably dopamine involved in there somewhere. It's the best buzz I've ever had.
Thank you both, and I think it shows there's different opinions and attitudes, and obviously environment and exposure does play a part. Graham, I know that an innovative mindset can be learned. I know it because I've learnt it, and I've been lucky to be inspired by both of you, Graham and Minas, and all of the conversations and the people before us. You know, me imagining, what if I was around 50 years ago when phaco was being developed, or the intraocular lens? I was around when endothelial keratoplasty started, and that's when I learned about epikeratophakia that you talked about. And these things that come before us and that we witness at the film festivals, which should never be missed, they inspire an innovative mindset.
I mean, it's a nice topic because we will disagree, and you can only talk personally. For me, I think I've got an innovative mind. It's not something that you want to do or desire to do, and it's not something that you can stop. It can be a burden because every day of your life, you're stimulated and detracted from new projects. There's a magazine called Make Magazine. Anybody know Make Magazine? So there's a subgroup of people who are called makers, and they even have their own magazine now. It's called Make Magazine, and it's full of people. I see myself as a gadget man, and yes, you just really have to choose which ones you explore very carefully, because every day you're thinking of new, and it's not driven by profit. It's not at all. And it's not even driven, I hate to say, by the betterment of humanity and mankind. It's something innate and something that you can't switch off.
I'm going to agree more with Jackie, actually. I think that, everyone has ideas that come in their mind. I think that what Minas and Graham have done is they've had those ideas, and they've had the discipline and the perseverance to develop them. I mean, put your hand up if you've had an idea that you thought, hey, this might be, a good thing to do, and then didn't actually sort of continue with that. And then a year or two later, you sort of see that someone else has done it and actually taken the trouble to do it. So I agree with you that everyone has the capacity for innovation. It's not the coming up with the ideas. It's actually being able to take them forward. And that's the point of this symposium, is to try to encourage more people who have those ideas to get in that mindset that Minas and Graham have, and to be able to sort of take those things forward, because it's going to be better for our patients in the long run. You may have an innate capacity, but we all have the capacity for ideas, I think. I think you're right, Jackie.
I think we might move on to the next speaker, Jared.
Okay, it gives me great pleasure to introduce Jill Hopkins, who you saw just on the video there. Jill is an ophthalmologist and retinal specialist who spent 20 years in clinical medicine and academic research, and importantly, over 12 years in industry in various stages of research development. She has expertise in hereditary retinal degenerations. She was an assistant professor at Keck School of Medicine, and she recently took on the role of Senior Vice President and Global Head of Ophthalmology at Novartis, leading a portfolio of drug, device and digital development. And before that, she was Global Head of Personalised Healthcare in Ophthalmology at Roche Genentech. So significant experience both in ophthalmology, in development of products, and in that critical role in terms of dealing with ophthalmology and bringing things to the market. So Jill, thank you very much for your time today.
Pleasure. Thanks, Jared. Can everyone hear me okay? And I think AV was going to run the slides for me from the room there. But pleasure to be here. I know we've had this symposium in mind for a couple of years now. It was halted by COVID on a couple of occasions. So lovely to be here and sharing the podium with some just outstanding speakers and insights on innovation. So Jared gave me this title of Lost in Translation, which I put a more positive spin, and we'll try and focus on some of the perspectives on how not to be. And if you go to the next slide, as Jared said, I've been very fortunate to have 3 decades in ophthalmology now, 20 of which were spent in clinical and academic medicine and research in both academic institutions and private practice in Canada, London, and the United States. And then just coming up 12 years now on industry in large pharma, small biotech, and doing a lot of data, digital, and AI as well. So today I'm really sharing opinions that are solely mine. I think it's really about sharing the learnings, the thoughts I've had, and I've loved how the themes have come together even from our first 2 speakers on those sort of kernels that keep showing up when we talk about innovation and bringing transformation to patients or healthcare systems around the globe. So I look forward to sharing some of those with you. On the next slide, Jared opened the symposium very well with the why, and we've heard that as well from Graham and from Minas thus far, that the why is really the critical piece. So I'm going to use 2 case studies that I've been closely involved in over decades now, that really show a couple of things around what Jared opened with. So why it's important for patient care. And I think what's exciting as we look at the future and the things we can start to bring to play and innovation in medicine in particular, always the patient and the problem at the center. That I think guides us. But we're also at this fantastic intersection where we're now seeing medicine, technology, compute power, data, engineering, all of these coming together in ways that really form that network of innovation. The same type of network that led Thomas Edison to be the one we now credit with the light bulb, because he had the infrastructure around him to actually deliver that and light up Pearl Street in Manhattan all those years ago. And although he did, Jared, make those workers perspire, he was also the first one to give equity to his workers. So he was innovative in that sense as well. We've talked about the mindset, and I'll share that as I go through, but it's really that yes and mindset. And this is what they talk about in improvisation. Actors who are thrown anything that might be thrown at them, they have to say yes and then they have to build that forward. And then that creativity is fun. It is fun. It's also difficult and time consuming. So we'll use these cases to bring some of that to life and hopefully have some good discussion as well. So on the next slide, a couple of things. Again, I think these have all been pulled through beautifully. And we didn't really coordinate with the speakers on this, but I think the problem and the why, we've talked about that. I think to me, regardless of the institution we work in, and some of us work in very big, complex institutions, but getting that why, the problem, and putting urgency behind that, you can supersede what can sometimes be a lot of complex process without having to compromise good decision-making. That's been a key learning for me. Failing fast, we don't like to say we fail in medicine. It's not how we're sort of wired, but if we think about it in an ordered sequence, if we iterate quickly, have the next plan in place, we can do very well and move more quickly. Embracing serendipity, I think, again, sometimes it can be the most trivial finding that may land in front of you, and it may actually be the one that produces the solution. So just keeping that open mind. yes, and I've mentioned, and then connecting the dots. How do we build a tangled bank of different ideas, borrowing, building upon the ideas of those around us? On the next slide, a couple of great quotes around growing a culture of innovation. And again, I think you can use this regardless of where you are trying to innovate, whether it's your garage, your basement, a large company, a small biotech, in the midst of being a registrar. I think anything is really thinking about these new ideas that may not be accepted at first. And I think we've heard from Graham and Minas as well, the repeated attempts, the endless demonstrations, monotonous rehearsals. This requires courageous patience. And I think that's a wonderful term to show what is involved. We have that sort of eureka myth that, oh, it all happens all of a sudden. We know that it doesn't. It's a very long, arduous journey in many cases. And then Stephen Johnson, who writes extensively on innovation, has some wonderful books, but I think love his points around the patterns are simple, but followed together, they make for a whole that is wiser than the sum of its parts. So he talks about embracing serendipity, make generative mistakes, let others build on your ideas, borrow, recycle, and reinvent, build a tangled bank. I think those are things that when you embrace them in the moment of trying to build towards innovation, it can be very successful. On the next slide, I'll walk through case study one. So the port delivery system, if you go to the next slide, the problem statement, if you click for me, was very much around anti-VEGF therapy. So we had over a decade of anti-VEGF therapy delivered intravitreally around the globe. It had been immensely successful. It had actually reduced blindness from neovascular age-related macular degeneration by over 50% around the globe. So it was very successful. We knew from clinical trial outcomes, we anticipated wonderful increases in vision that we had never seen before. What we saw over time, though, was that the real world vision outcomes were not matching what we would have anticipated from clinical trials. And we saw that really based on ability for, be it from systems or patients or marketplaces to support the frequency of that intravitreal injection, it was deemed to be monthly to provide the best outcomes. We saw that vision was not reaching those levels and that we were leaving a lot of vision on the table. In some parts of the world, it looked like we didn't have therapies on market again. So this was concerning and was the host and catalyst for a massive amount of work in long-acting drug delivery. If you go to the next click, you'll see here Nancy Holekamp recently published the results of the Archway Phase 3 trial for the port delivery system in neovascular AMD. Lovely design of an implantable device that could be placed in the eye in the operating room in about a 15 to 20 minute procedure. This could then be refilled in the office. And the idea was that you could have extensive, long-acting duration of anti-VEGF therapy delivered through this with about 6 months of one-time surgery refill in the office as needed. Indwelling, designed to be there for the lifetime of the patient. So very innovative and exciting. And the next slide shows that, in fact, this worked. The phase 3 Archway trial showed that the port delivery system, when refilled every 6 months, had visual acuity and OCT findings that matched that of monthly intravitreal injections. So very exciting. Again, a wonderful intersection of so many different places in medicine, engineering, biology to bring this together. But if we go to the next slide, we see a true example of courageous patience, right? So if we look back to the first discovery of VEGF back in 1989 with Napoleon Ferrara at Genentech, it took many, many years to get ranibizumab to market as an intravitreal agent approved by the FDA in 2006. Dr. Jean Dewan, who is an innovator in many areas around the space of retina, founded Foresight Vision 4 shortly thereafter, started a phase 1 trial looking at the first prototype of the port delivery system in humans, and Genentech very interested in having something like that to support its portfolio of intraocular medicines, licensed with them in 2010. We then we completed the phase 1 trial in 2014, and in 2015 started a phase 2 trial. But we did have a host of challenges, many of which are nicely detailed in the paper that's referenced here by Tony Adamus and Jean Dewan that really highlights all the details around this. But we had issues with vitreous hemorrhage that were being seen very early in the post-operative period at a rate that wasn't acceptable. So we had to pause the trial and quickly iterate on how would we come to find the solution for this. Because we saw the hemorrhages on day one post-op, we were convinced it was a surgical fixable situation, but we had to move very quickly. And the best example of serendipity that I have is that while working through this, we had the cases coming in clinically. We'd put a hold so that we could ensure patients were not continuing to experience these. But we had a wrap up of some of our early animal work. Our preclinical team was presenting just to close out the mini pig data that they had done to work to get us to first in human. And they were flipping through the slides, we were going through it, and they commented on these vitreous hemorrhages that were self-limited and disappeared. And they were about to move to the next slide when I said, wait, go back. And we then pulled together very quickly an animal model in the mini pig that we could iterate on literally day to day to weekend to come up with the solution, which was in fact to laser the choroid before putting the insertion in. The vitreous hemorrhage rate decreased, and we were able to complete phase 2, start phase 3, and get to approval last year, 2021, with the port delivery system. So again, huge amounts of effort over many, many years. And again, just that intersection, that bringing together of engineers, preclinical, clinical, to see the problem, solve the problem, and move forward. Again, not quickly, but with a good iterative approach, ultimately bringing transformative therapies to patients. So a very exciting piece to be involved in. On the next slide, I'll touch on something quite different. So the Insight Health Data Research Hub, which I had the privilege to be involved in, again, solving a different problem and very much looking at the future of medicine, healthcare, and how we'll look at solving problems. So if we go to the next slide, what we were interested in evaluating and solving for with the Insight Hub was that, we are seeing more and more the artificial intelligence and big data story really starting to unlock insights we've never before deemed possible. And yet we're not quite at the point of having been able to translate this into clinical care or decision-making. Vast amounts of routinely collected data are pulled in every day, and yet how do we use these, harness them, refine them to actually further research and development, and most importantly, support breakthrough in eye disease and other areas. And key issues to overcome are data quality, curation, access, patient privacy, all of these things we're thinking about in the world of actually bringing AI to everyday use for our patients. So we go to the next slide. I think, again, speaking of a tangled bank, this is an article that I wrote with Pierce Keane and Konstantinos Balaskas from Moorfields, who are also very much involved in the Insight Hub. But we were really thinking about, much like drug development, if you're going to bring an artificial intelligence solution from ideation to adoption, it has multiple stages that it must go through with multiple key stakeholders involved at every step of the way. And what you see really across that second light blue bar at the top, data drives all of that. So we saw that for every stage, having access to well-curated, highly understood data sets was going to be critical. So if you go to the next slide, we convened a group of people, and we'll talk about that a little bit more, that network of innovation. But what was important, although we came from very different backgrounds and had different motivations, but perhaps at an individual level, what we were very interested in doing at the higher level, the sort of North Star that brought us all together, was how do we harness that power and really have short and long-term impact for patients, physicians, and systems. And when you look at, the paradigm that might allow us to predict progression of disease, for example, or response to therapy, something we haven't been able to do well in the past. Could we actually enable over time prevention paradigms? Could we start to see that transition from health to disease and intervene in a different way that we hadn't before? And the concept of harnessing this ongoing data collection for dynamic data sets could be really intriguing. And with the next slide, you'll see the group that we brought together, which again, vastly different backgrounds. We had Action Against AMD, which is a patient charity based out of the UK, Google with their fantastic AI and cloud-based knowledge. We had outstanding clinicians from Birmingham and from Moorfields Eye Hospital, where again, they had data sets that were large, collected over many, many years. And we brought that together with Roche, who was interested in the personalized healthcare idea between how do we use data, digital AI to drive more efficient drug development and better outcomes for patients. So it really was an interesting collective. And again, my point about sort of being able to harness that power and build toward decision making in a very quick, I mean, you look at institutions like that and think, oh, that would be hard to do quickly. We came together, wrote the grant, and had a submission within about a month, which we were then very fortunate to get the grant through the HDR UK. So there's a wonderful website that outlines now where the project is at. The data sets are available and researchers can access them. They cover everything from glaucoma to retina to cataract to dry eye. So I think really worth a look there at what that came. Again, courageous patience to get it there. Was a lot of work over the last 3 to 4 years, but tremendous to see that coming to fruition. And on the next slide, the Insight Hub itself highlights, and there's a video on YouTube where a lot of the founding partners talk about this, but it really was the concept of bringing together this expertise. And much like that tangled bank that Stephen Johnson refers to, we really start to, in this combined expertise model, we can innovate and progress in a way that no single organization, public or private, could achieve on its own. And I think that's been really, for me, a learning, whether it be in a port delivery and a data the other we've heard from our other speakers, just that how we bring that together, the expertise we can leverage, and building that mindset, that team that really can deliver on the problem at hand. On the next slide, we'll just close with how not to be lost in translation. I think we've again highlighted this sort of iteration, embracing the change, having the problem, recognizing that with urgency. And I think what's really exciting is keeping that vision of what you're trying to do in terms of changing the paradigm. And to do that, you've got to share experiences and support needs. Communication is key. Always thinking about best possible outcomes for, is it patient, physician, system, whatever that may be that you're trying to achieve, but really focusing on that transformative impact to get you through the long nights and days, to have your family still talk to you at the end of the weekend when you've been working. I think it is a really exciting place to be. So with that, I'll close with just sort of the final thoughts around, if we go to the next slide, and if you click again for me, sort of think about that tangled bank and how you can really listen and pull that together. The next click really shows that improv mindset, the yes and, right? And I think it's really something that, again, when you surround yourself with people that have that mindset, that motivation, this just happens. And that's a really exciting place to be. We've heard that from Graham and Minas as well. And then on the last slide, the communication piece, don't leave people unclear about what you've said and what you're trying to achieve. And with that, I'll say thank you.
Thank you, Jill. We'll have a little bit more discussion after our next speaker, who is Andrew Batty. He's an experienced medtech professional and has served as the CEO or director of public and private companies. And he's had tenures at large pharma, startup biotechs, and he's worked along VCs, and he's been part of raising capital and spin out for more than 20 new technologies. He's got a proven track record in R&D collaboration, and Andrew's going to talk to us about how we get that information to a product. Thanks, Andrew.
Thanks, Jared, for the invitation to speak. Jared picked up on one of my lines, which was a throwaway line in one of my presentations to some academic researchers, where I was encouraging them to take the next step through to commercialise their innovation. So I hope that doesn't offend anyone in the crowd. My support in innovation, translation and commercialisation comes from, as Jared said, 25 years in medtech and biotech. Some have been really successful and we've enjoyed the process and they've ended in champagne. Others have died a miserable death and we've stuck a fork in them. So, but I think one of the themes that's come out is that it's always fun and you've got to enjoy it. And I'd encourage people to do that wherever they are on that innovation spectrum. So if you're creating an innovation or whether you're translating that innovation through to an invention, I generally work in that back end. So I let the idea generators kind of appear to my right here. I let them play in the sandbox, and then I try and grab hold of those good innovations, translate them through to an invention. So I hope that gives you some perspective. So clicker. Sorry. So some of the projects that I'm involved with, 3 ophthalmic projects. Bienco's a consortium of university and research institutes
that have united to create a multi-layered bioengineered cornea. That's currently in R&D phase, concept phase, and it's seeking MRFF funding. iFix Medical is a privately funded startup company with Jared, and we've licensed technology out of the University of Sydney, and we're creating a topical medicine for the treatment of corneal disease, corneal trauma and ulceration. That's at large animal stage. Vivid White has developed a microfluidic internal resisting glaucoma drainage implant that's currently performing magnificently well. If there's any investors in the room, please don't think that that's a forward-looking statement. It's in a 10-patient human study with Michael Coote down in Melbourne. And one other really cool technology that I'm working on is cerebral therapeutics. So that's they've developed a direct-to-ventricles drug delivery system, and the next stage is to try and then deliver drug on cue. So this is where I implore everyone to get involved in some form of research, innovation, product development or commercialisation. One of Jared's favourite lines that I sort of have used as a throwaway, but an experiment in a laboratory will not cure blindness. It needs to be translated into clinical practice to impact humans. So we need to translate research from the publicly funded research institutes and encourage investors to take the punt and create it. And it's up to us then to create compelling data that potentially de-risks that project, that encourages them to take the punt. Then the medtech companies, the Jills, where Jill's from, can then acquire the technology, put it into large-scale manufacturing, and at that point then we can actually impact humans. So a publication is interesting, right? It's pretty exciting, but a commercialised device or drug can save lives and cure blindness, as to Jared's point. So there's a real push nowadays for, and this is part of the theme here as part of getting closer to identifying the actual need. And there's a real push nowadays for facilities and institutes such as ACMD is down in Melbourne. It's located on St Vincent's Hospital site, Translational Research Institute up here in Queensland, and there's lots of others. But they're really trying to work more closely with the clinicians to identify that real problem, the unmet clinical need. Throw in some research and you get the invention, right? It's at that point there where, this is a slide I picked up, I've sort of plagiarised, bastardised it from MIT actually. But it's at that point there you get your invention. Once we've identified the problem, initiated the research, you then translate and commercialise and to create an actual product. That's when we actually create the innovation, and that's what I implore everyone to take that next step. Now, along that trajectory, at any point you can get involved. My special skill is in the latter phase. I try and search out the invention. I try and find people to my right, people like you who have come up with great ideas. Yeah. It's really important that at that concept phase you start fleshing out and productise. I like to call it productising. Well, they're really defining the product features of an innovation. And this is an example for iFix Medical for a drug delivery or a bioink delivery product. But there's lots of examples where potentially we've progressed with product development and we really haven't conceptualised what the product is enough, and that is how it's going to be used in the theatre, how it's going to be delivered, how it's going to be stored on the shelf, how it's going to be mixed, etc. So, in that ideation phase or developing the concept, it's great to start thinking and start prototyping, but really start conceptualise how that product is going to be used and sit on the shelf. So this is where I implore you to get a commercial person involved early, and it sort of harks back to that earlier slide with Darth Vader. They don't really call it the dark side anymore. We encourage you to connect with industry. And you can see from this side, if we consider the stages from idea creation all the way down to feasibility through to launch, you can see there's a significant amount of work required along the way where commercial people need to get involved. And I'd encourage everyone to do a business case. It's not a business plan. Business plan is something that sets out a document that says what you're going to do for the next 5 to 7 years, how you're going to commercialise. A business case is all about looking at, can this thing actually be made? Is it viable? Do we have the resources? Do we have the skills? Can we manufacture it at a viable price to then eventually sell it? You know, something to displace a Band-Aid is going to be pretty hard, especially if you're trying to sell it for $30 a unit. So when we're looking at technology to invest time and money into, we generally look at 3 key components. We generally look at the market, we look at the IP position, and then the product performance. A lot of people initially start talking about product performance very early. So if you start thinking about the market assessment, what's the immediate unmet clinical need? I implore you to validate that with your colleagues and with the hospitals, with the other users, because what you might think is a great idea, don't mean to offend anyone, but it might not actually get widespread traction. So validate the concept very early. If you can't answer who's going to buy this and why within 20 seconds, don't do it. You've got to be able to think there's got to be an economic reason. Trying to get stuff into the hospitals now and try and sell stuff without a health economic benefit or an argument is really challenging. So it's got to go beyond than just the Medicare or the MBS item codes or the prosthesis schedule. You've got to really understand, why is this thing adding value? Be realistic when you're assessing the market size. For example, a glaucoma product, your market is not every blind person in the world. Your market is not prescription medicine, $10 billion prescription medicine market for pharmaceuticals for glaucoma. Your market for a glaucoma MIG device might be specifically those patients that get immediate benefit from it right now. So you see so many presentations and people say, that their technology is solving a such greater or applicable to such greater market, and I encourage you to really focus on your market need or the market size. Understand your competitors, how crowded, and venture capital firms and investors look at this, because they first look at the market size and then they try and determine based on what sort of section of the pie they can actually secure. And then from that, they can develop a valuation. Part of that valuation depends on how good your IP is. And to get a patent, I think Minas mentioned about developing a great relationship with a patent attorney. I would say try and find, this is my little tip, don't follow firms, patent attorney firms. If you're at a university and they have a deal with ABC patent attorney firms, ignore it. Try and find a specific patent attorney with a skill in the area that you're working in, and follow individual patent attorneys. Don't follow a firm. That's my little belief there. For a patent to get up, it's got to be both novel and have an inventive step. The good patent will probably just allow you to play in the market, so that's freedom to operate, or put your towel on the beach, so to speak. A better patent allows you, maybe it protects you a larger portion of the beach, and remembering a venture capital or investor or a pharma company acquiring your technology is going to value your technology accordingly, depending on how much of the market they can secure. But the best patent are those where you got the whole beach to yourself. You're basically first to market, a new segment, something you can protect others and prevent others from entering. It's got to be commercially enforceable. I mean, if you've developed and patented a method to hold a scalpel a certain way, it might be novel, it might be inventive, but it's never going to be enforceable. You can't actually enforce it. So, and also consider nowadays with cloud computing, a lot of algorithms in the cloud. Jill alluded to, database algorithms put in the cloud. Then you don't actually need a patent. You might have a business model or a transaction model that subscribers can access IP that way. So the last one was product performance, and that was, does your product work as promised? We then look at that, look at the clinical data, the preclinical data, and critically, is it really as unique as what you say? So in summary, I'd encourage you to translate and commercialise your research and validate your unmet need, check it out with your colleagues and make sure that your idea is not kind of good amongst your household, that it is actually valid across the board. Get commercial people involved early and define the product and the characteristics. Make sure you really define how it's going to be used, where it's going to be used. Propose a business model. That might just be a simple, that might be very, very easy if it's a straight medical device or a pharmaceutical good. Understand the reimbursement route with computing and software. Often devices now are very different. Is it a capital equipment item or is it a consumable? And don't focus too much on the exit. All the venture capital firms talk about, having an exit strategy. My suggestion is don't worry about that. Focus on developing a business or a viable product, right? And then the exit strategy will fall into place, because we know there are 2 or 3 or 4 along the way. They're pretty obvious, and so I would suggest not to focus on that. Thanks.
Thank you, Andrew, and thank you, Jill. So many amazing points to touch on, and we don't have a lot of time left, but just a couple of points. Both of Andrew and Jill touched on failure, and I think we won't make this our last point because it's a downer, but there's just no entrepreneur and no innovator who hasn't had ideas that have failed. And Jill gave a good example of the port trial being paused, and Andrew, you talked about some solutions, niching down, validating your idea, presenting a business case. But how do we deal with this in medicine when failure can really disrupt ideas because of safety? And do you think that regulation and restrictions have made innovation more difficult in medicine?
Go for it, Jill.
Sure. Thanks, Andrew. It's a great question, Jackie. I think that, listening to you, Andrew, I think you had a lot of really good points about early engagement, right? And I think that that's true for the regulators as well, right? To early conversations with them about your plans. Certainly when we had the issues that came up, we were talking all the time with the regulators about what our plan was, how long we would look at trying to solve that, what the clear path forward was there. So I think that regular interchange and exchange is critical. Certainly the regulators talk about being happy to have those early engagements and discussions, and then it's an ongoing dialogue. So I think that's a key piece. And I think, the regulations are there for a reason, right? I mean, all of us have to have patient safety at the heart of what we do, right? We hope for obviously better outcomes and transformative things, but if it doesn't turn out to be a safe path forward for them, that has to be, the reason it stops, right? And I think that the regulators are there. I think certainly in the space of, data, AI, when we look at trying to build that's an interesting area, right? Because I think that we're all learning together of what is the development path for an algorithm, for example, what will be required, how do you manage safety there? And most regulatory agencies have come out with good guidelines at the moment on what that is. But I think they're also looking to us to have more dialogue and say, does that make sense going forward? So I think, as always, it's that yes and how do we sit down together and think about how to go forward.
Yeah, I agree, Jill. And the TGA really don't bite. They're actually fantastic. So when you're developing a drug, they're actually looking towards you, or developing a device. They're looking at you to flag the potential risks. Because if you're an ophthalmologist, you know the risks associated with your device better than anyone. So there's going to be the process of device development, getting a device to market, is all about design control, and that is about designing what the product is, identifying those risks, mitigating those risks, and being able to then show that you've mitigated those risks. So as much of a way, as Jill said, that engagement with the regulator is really important, because they're actually looking towards your feedback and making sure that you have adhered to a traditional process.
Thank you. That's another great, I know it was a throwaway line, but the TGA doesn't bite is going to come up again. Collaboration's another theme that really came up in both of the last two talks. And Jill, you taught me a new term, the network of innovation, and I love that. And I can see that it's really important. Could you speak to new ideas? And I know I've personally been both ways. You can have an idea and think, oh, I'd better not tell anybody else. But actually, once you do start telling people, you develop a network, the idea gets better, and you can actually do something with it. How important is collaboration when we're talking about innovation?
I think it's key, Jackie, and especially as the world gets more complex and expertise in certain domain areas gets deeper and deeper and deeper, right? So not any one of us, be it an individual, an organization, an academic institution, can drive what it takes nowadays to actually deliver on that, right? So I think that collaboration is key. And what I've found interesting, the sort of longer you're in the field and the more people you know, you really do start to build that network of like-minded people, and you know who you can call to bounce it off. Or, you know, Andrew, you and I have had a lot of good discussions since we've been linked up over a couple things, and it's just you just start to build that idea. And sometimes you'll sort of cold call and say, can I talk this, through with you, and just start to develop that. So I think it's critical. And again, it's all about, I think we used to feel like, oh, I'll be the most knowledgeable about this or the smartest. And it's really not that now. For me, I think success is, how do I get the right people around the table to make this happen? And it's, as I say, it's not a one person, one organization win anymore. It's got to be that collaborative mindset.
And you need to bring, as Jill said, you need to bring people along on that trajectory. So you've got to open up and share your ideas. Just make sure your patents are tight. Make sure you've got your IP in place, and then you can share the love with as many as you like. So you need to. Just on one point there, quite a lot we see clinicians or researchers or universities, publicly funded research institutes, go to the media. Now, my advice there is always make sure you've got a genuine reason to go to the media. So it might be to create awareness for investment. It might be to drive patients into a clinical trial. But push back on your universities or the research institutes if they want you to promote your device, because you're only new once. So make sure you use the media really wisely. Collaborate with who you need to along the way, but when you want to go to the media, you structure it and make sure it's part of your plan to secure funding or to get patients or create awareness.
Okay, thank you, Andrew. Look, we've run out of time, but I just wanted to thank the panel, and I think it's been really insightful. I've learned a lot. We didn't want to make it scary, but it did sort of sound hard, sort of taking idea to market, but there is a pathway is the important thing. And as I said at the beginning, the point of this symposium is to encourage, as Jill has articulated very nicely, that network of innovation. And we want to see that. And I think it is really important that there are going to be some new Minas Cronios and Graham Barrett coming through. So thank you all, and thank the audience for hanging around. Appreciate it very much.